Very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible? And also please let me know if you can see the video and the presentation. Let me know if everything is fine with the audio and the video and the presentation. Okay, thank you so much for confirming. We will wait till it's 6 p.m. and more students join, then we will start today's session. So good evening to the students who have joined. Please wait till it's 6 p.m. and then we will start today's class. Meanwhile, let me know if you can hear my voice properly and if you can see the presentation and also the video. I have kept the microphones off for the students so that there is no disturbance in the class, but the chat box is always open, so you can always communicate through the chat box. Since I am recording the session, I have kept the microphones off, but you can communicate through the chat box. We will start shortly, so please wait. call open so that students can join according to their time. Okay, so I hope my voice is audible, right? So let's start with the introductions. Meanwhile, when more students join, they can automatically get into the class. So I will start with the introductions at first, and then we will start today's class. So I think my voice is audible and you can see the presentation. If not, please let me know in the chat box. Okay, if there is any problem with my voice or the presentation, let me know in the chat box. So very good evening to all of you. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is our week 3 live session. So previously we had 2 live sessions already and we have completed I think around 3 weeks of the course, and today we will discuss about the topics which are in the week 4. So right now your week 4 of this organic chemistry in biology and drug development course is live, and all the course content, the videos are live, and also you have given the assignment for the week 4 which you have to submit in a due course of time. So today what we will do is we will discuss the week 4 course material, and we will discuss the type of questions which are given in the assignment, and also these type of questions solving will help you not only for the assignment but also for solving the for understanding the course better and for your final examination too. Before starting, I will try to tell you few points which I state in before every class. So there will be a problem solving session every week, and this session will be on every Monday from 6 p.m. to 8 p.m. and these sessions are live on Google Meet platform. So if any network disruptions happen from my side, if there is any network disruption happen, so please let me know. I will try to rejoin as soon as possible, but do not leave the session. Okay, I will join as soon as possible, and if the network problem is for your side, you can rejoin at any time, there is no problem. And since I am recording this session, so you don't have to worry about missing out anything due to the poor network connection, because I will share this recording and also the PPT with you in the due course of time after the class. As soon as possible I will share these things, and during the session students can post their question and doubts to the chat box. Since I am recording the session, I have kept your microphones and cameras off so that there is less data consumption, and also so that I can record the session properly. And but the chat box is open always. I am monitoring the chat box every time, so if you have any doubt, any query, anything, please let me know in the chat box. If there is any problem listening to my voice or seeing the PPT, let me know then only. And if still any doubt persists after class, just you can drop an email to me. I will share my email ID at the end of the session. You can you can send an email to me stating your doubts. I will try to answer your query, and this presentation and class recording, as I have already said, it will be shared to you after the class. So before starting, let me check how many of students are joined. So I hope my voice is audible to you, and you can see the presentation, right? If there is any problem, let me know. Okay, I hope it's fine. Then I will proceed with the course material what is there in the today's class. And like every day, I will show you around 20 MCQ questions. Each MCQ question will be given, I will giving 4 options for each question. You have to identify what is the correct answer and let me know in the chat box. Like for every question A, B, C or D, which of the following is the correct one, you have to identify and let me know in the chat box. Okay. So let us see what is, so as I have already stated that today it is a week 3 live session where we will cover the week 4 course content. Right now the week 4 is live. So what is in this week 4? The week 4 has the enzyme kinetics which is continued from our last week. Also we have discussed little bit about enzyme kinetics, the basics ones, but today we will discuss mostly about the enzyme kinetics, the advanced kinetics, and the enzyme inhibitions, and the concept of enzyme inhibition. It will continue for a long time. We will discuss different type of inhibition, different graphs associated with the inhibitions, and also we will solve a lots of mathematical questions in today's class. And the problems, after solving the problems, we will discuss little bit about synthetic biology also in today's class. Okay. So right now I am starting with the question answer session. So if there is any problem till now, let me know. If not, I will continue. I hope it's fine, right? My voice and the presentation, everything. So I am starting with the session. As I am showing you a particular question, try to answer the question. Okay, there is no marks answering, so if your answer is wrong, it doesn't matter, but participation is very important. So I request all of you to participate and answer the questions. Whatever you think is the correct answer, let me know. Okay, so I will start with the question number 1. So the problem 1 of today's session is, the active site of the chymotrypsin consists of a catalytic triad of which of the following amino acid residues? Your options are: number A, serine, histidine, arginine; number B, serine, histidine, glutamate; number C, threonine, histidine, aspartate; and number D, methionine, histidine, glutamate. So which of the following 3 amino acids are the catalytic triad of the active site of chymotrypsin? Chymotrypsin we have discussed, I think, in the first week, that it is a very specific enzyme for the enzyme cleavage, and the particular active site of the enzyme has a particular 3 set of amino acid residues which are known as catalytic triad. You have to identify this catalytic triad of the active site of the chymotrypsin. Your options are as given: number A, serine, histidine, aspartate; number B, serine, histidine, glutamate; number C, threonine, histidine, aspartate; and number D, methionine, histidine, glutamate. I will give you some time, think, and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 1. So what is the correct answer? What is the catalytic triad of the chymotrypsin enzyme? Think. I will give you some time and then we will discuss it. Okay, let me check the chat box. I think I will have few answers at least. Okay, I have got only one answer which says it is number A. Okay, let me check if number A is the correct one or not. Others also please participate. Okay, it's only for your understanding of the course better, but if you do not participate, it won't be any fruitful for this class, right? So the active site of chymotrypsin consists of the catalytic triad of the amino acid residues. The correct answer will be, very good, it is number A, serine, histidine and aspartate. So serine, histidine and aspartate are the 3 amino acid residues which are known as the catalytic triad in the active site of chymotrypsin. So the catalytic triad, the 3 amino acid residues in the active site in a hydrogen bond network. So, this is known as the serine, the residue number 195, histidine, the residue number 57, and aspartate, the residue number 102. These are essential for the effective catalytic activity in chymotrypsin. Catalytic triad action converts OH group of serine 195 into a potent nucleophile. So, what does it do? Basically, this is a catalytic triad. Okay. So, here it is the aspartate 102 residue, here it is the histidine residue 57, and here it is a serine residue. So, what does this catalytic triad basically do is, aspartate residue has a negative charge, right. So, this negative carboxylate ion takes the proton from this imidazole ring of the histidine, and in subsequently what does it do? This imidazole ring, it takes up the hydrogen, or it takes up the electron density from this serine 195. So, this hydroxyl now converted to a alkoxide, which is a more potent nucleophile than the normal hydroxyl in the serine 195. So, that is how this hydrogen bonding network has enhanced the nucleophilic power of the serine 195, which is very crucial for the activity of the chymotrypsin enzyme. So, the tetrahedral intermediate of the serine protease mechanism. So, basically these proteases, we have already discussed that chymotrypsin is a particular protease which cleaves the amino acid residues and the particular protein chain. Okay. So, these are called the serine protease, where the serine is the particular amino acid which takes part in the attack of the as a nucleophilic attack. So, this tetrahedral intermediate of the serine protease mechanism, aspartate 102 stabilizes the positive charge acquired by the histidine 57 and the negative charge on the carbonyl oxygen of the P1. Okay. This is the P1, this proton 1, the protein 1. So, residue of the substrate is the stabilized by the main chain amides of the enzyme that comprise the oxyanion whole. So, basically you have to remember this thing, that in the case of chymotrypsin there are 3 amino acids, aspartate 102, histidine 57, serine 195, which are crucial for this activity, and serine is the amino acid by which this particular chymotrypsin enzyme attacks the substrate. Okay. And this is known as the catalytic triad, and this catalytic triad works through the hydrogen bonding network. Okay. So, do you have any problem understanding the question? After each and every question, let me know that if you have understood that thing, or if you have any doubt for that particular question. If you have doubt in a particular question, I will try to repeat it, and if it is okay, then we will move forward. So, is it clear, or you have doubt? Let me know. Whenever there is a doubt arises, let me know in the chat box. I will repeat that particular question and we will discuss it a little bit before moving toward the next question. So, I am moving toward the next problem, problem number 2. If you have any doubt, you can let me know in the chat box. Okay. So, problem number 2, it says that which of the following diagrams best describes the equilibrium associated with a competitive inhibitor. Here E is the enzyme, S is the substrate, P is the product, and I is the inhibitor. Your options are number A, E plus S. This is in the equilibrium with ES, which is enzyme substrate complex, which gives us the enzyme and the product. Here we have the inhibitor which binds with the enzyme, and then the substrate, which is the enzyme inhibitor substrate complex. This is the first one. The similar here, number B, it says that enzyme and substrate gives us enzyme substrate complex which is bind with the inhibitor. Number C is the enzyme is binding with the inhibitor only, not the enzyme substrate complex or anything. And number D, here the substrate is binding with is in equilibrium with the inhibitor. So, which of the following diagram best describes the equilibrium associated with a competitive inhibitor? A, B, C or D? Look at the diagram very carefully, think what is a competitive inhibitor is, and then answer the question. I will give you some time, think and let me know in the chat box which of the following is the correct answer for the problem number 2. Which of the following diagrams best describes the equilibrium associated with a competitive inhibitor? which of the following diagrams best describe the equilibrium associated with a competitive inhibitor? Here E stands for enzyme, S for substrate, P for product and I for inhibitor. Let me check the chat box now. Let's see if there are any answers. Where can we have the recorded sessions? Okay, first let me see the answers. Number A, number B, number A. So I have three answers, 2 number A and 1 number B. I will check the answer, but first let me show you where you can find the recorded sessions. So I think all of you have this organic chemistry, you have associated with the SWAM portal, right? All of you have this announcement portion where you have the courses and the lectures, all the lectures and the quiz, and here you will find there is a download video option, text transcript, books and problem solving session. If you click on the problem solving session part, what you will find, there are 2 options, there are problem solving session recording and survey. If you click on the problem solving session recording, so it will take you to this kind of thing. Here written here is click here. If you click on this portion, so you will find here is my ID and here is the folder link. If you go to this Google Drive folder, so I will show you once. If you go to this Google Drive folder, you will find the week PPTs, right? Here is a week 1, here is a week 2. These are the last week 2 weeks PPT which I have already uploaded. Here from here you can download the PPTs and you can read, and also there are YouTube links for the class recordings. You can go to the link and watch the live sessions, the recorded session, if you miss anything in any case. Okay. So that's how you have to download the PPT, and also you have to check the recording. Okay. I hope it is clear where to find the recorded session and where to download the PPT. If it is not clear, I will again I will tell you at the end of the session. Okay. So right now let's focus on the problem number 2. So here it is given 2A and 1 number B. Now let me check which of the following is the correct one. So the correct answer is number C. Number C is the correct diagram which describes the equilibrium associated with a competitive inhibitor. So now let us first understand what is a competitive inhibitor. So basically a competitive inhibitor has roughly the same shape as the substrate, so it can fit into the active site of the enzyme. The competitive inhibitor is often product of an enzyme catalyzed reaction. When a lot of the product has been made, the product would compete with the substrate for the enzyme's active site, showing the reaction down. A competitive inhibitor is able to fit at the active site, and when it is there, the substrate is unable to bind with the enzyme and the desired reaction does not occur. The inhibitor and substrate both compete for the active site. Okay. So this is an enzyme. We all know every enzyme has an active site, and that particular active site substrates bind. So competitive inhibitor is those inhibitor which bind to the active site of the enzyme in the place of the substrate. Okay. Why it is called competitive? Because it is competing with the substrate for binding to the enzyme's active site. So now if you see in the option number C, you can see enzyme is binding with the inhibitor and it is forming an enzyme inhibitor complex, just same as the enzyme substrate complex. So that is the equilibrium which is particularly for the competitive inhibitor. So generally normal enzyme maximum rate of the reaction can be reached, and when the line plateaus are all the reaction occupied with their substrate. Competitive inhibitor, remember the competitive inhibitor competes for the active site with the substrate. When the substrate concentration exceeds the inhibitor concentration, the reaction will proceed and maximum rate of the reaction can be achieved. Non-competitive inhibitor, so now we have different type of enzyme inhibition. I will slowly move toward the other inhibitions also. So first we have learned about the competitive, and now let us discuss about the non-competitive a little bit. So these inhibitors don't compete for the active site. Just as you can see, the competitive inhibitor is binding to the active site of enzyme, but the non-competitive inhibitor, they do not compete for the active site. They attach to an alternative site of the enzyme, changing the shape of the active site, preventing the substrate from the binding. Non-competitive inhibition cannot be overcome by increase the substrate concentration, therefore maximum rate of the reaction will not be achieved. Regardless of the substrate concentration, this inhibition lowers the amount of the usable enzyme. So, now you can understand what happens when there is a competitive inhibitor. It binds where the substrate binds. So, if you use the lots of the substrate concentration is increased, what will happen? The more the substrate are there, the more the possibility of binding to the substrate with the enzyme is higher. So, we can achieve the highest enzyme activity. But if there is a non-competitive inhibitor, it will not bind to the active site, it will bind to a different site of the enzyme, and what it will do, it will alter the active site. So, now if the active site is altered, what will happen? The substrate cannot bind. Even if we increase the substrate concentration, it will not improve the enzyme, the rate of the enzyme catalyzed reaction. So, that is why the non-competitive inhibitors are also the irreversible inhibitors. Okay. So, these are the competitive and non-competitive inhibitor. Now, let us go to the next question. So, I hope it is clear. If there is any problem regarding competitive inhibitor, let me know. Let me know if there is any problem. Okay, I will rephrase the question and I will discuss it again. So, I hope it is clear. Then we can discuss that. Go to the next question. So, the problem number 3 says, what is the mechanism of action of orlistat or an anti-obesity drug? Your options are: number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, and number D, irreversible inhibition. So, what is the mechanism of action of orlistat, an anti-obesity drug? Try to answer it in the chat box. I will give you some time, and then I will show you the correct answer and we will discuss it a little bit. What is the mechanism of action of orlistat, an anti-obesity drug? What is the mechanism of action of orlistat? It is an anti-obesity drug. Let me check the chat box. Okay, I have few answers. Number D is the given answer. Okay, let me check if number D is the correct one or not for the mechanism of action of orlistat. So, the correct answer will be very good, number D, irreversible inhibition. So, irreversible inhibition for the treatment of obesity is a very famous example is this orlistat. So, fat in the diet is composed mainly of triglyceride, which are digested in the small intestine to fatty acids and to monoglyceride. The digestion products are then absorbed and act as a building blocks for the fat biosynthesis in the body. The enzyme pancreatic lipase is responsible for catalyzing the digestion of fats. So, inhibition of this enzyme will result in the reduced absorption of glyceride and fatty acids from the gut. Consequently, less fat will be synthesized in our body. So, orlistat is an anti-obesity drug that acts as an irreversible inhibitor of pancreatic lipase as a result of the presence of the electrophilic 4-membered lactone group. This acylates a serine residue in the active site, which is a part of catalytic triad of serine, histidine and aspartic acid. Okay. So, as you can see, similar to the here also in the particular enzyme pancreatic lipase, which is responsible for the absorption of the glyceride and fatty acids from the gut. Okay. This enzyme active site has a catalytic triad of serine, histidine and aspartic acid, and here serine acts as a nucleophile. So, as you can see, what happens if you use this drug. So, it will react with this serine residue, and it will acylate this serine residue and bind with it. So, if it binds with this serine residue, what will happen? So, now this serine residue cannot act as a nucleophile, and this is the irreversible inhibition. Okay. So, irreversible enzyme inhibitors can form a covalent bond to a key amino acid in the active site and permanently block the affected enzyme. The most effective irreversible inhibitor are those that contain an electrophilic functional group X capable of reacting with a nucleophilic group present on the amino acid side chain. Invariably, the amino acid affected is either serine or cysteine, because these amino acids are often present in active sites and contain nucleophilic groups such as OH and SH respectively, that are involved in the mechanism of action of many enzyme catalyzed reaction. Electrophilic functional groups used in irreversible inhibitors include alkyl halides, epoxide, alpha beta unsaturated ketone or strained lactone and lactams. The highly toxic nerve agents contain electrophilic fluorophosphate groups and are irreversible inhibitor of mammalian enzyme. Irreversible enzyme inhibitors are not competitive inhibitors. So, this increasing the concentration of substrate will not reverse their action of inhibitor, as the inhibitors cannot be replaced by the from the active site. These can cause problems if the buildup of the particular substrate leads up to toxic side effect. For example, the monoamine oxidase inhibitors or MOAIs, okay, they block the metabolism of noradrenaline and have antidepressant activity. Unfortunately, the metabolism of substrate other than the noradrenaline is also inhibited, and leading to a buildup of these compounds and a serious side effect. More modern MAIs are now designed and reversible inhibitors in to avoid this problem. So, basically this is the irreversible inhibitors, and there are many I think medicinal medicines which use this particular irreversible inhibition mechanism to inhibit a particular enzyme and for a particular disease. Okay. So, the number D was the correct answer. Irreversible inhibition is responsible for the mechanism of action of all listed. Okay. So, now I will move toward the next problem. If you have any doubt, any problem, let me know. Okay. Problem number 4 says, which of the following inhibition is called mechanism based enzyme inhibition? Your options are: number A, competitive inhibition, number B, non-competitive inhibition, number C, suicide inhibition, and number D, active site directed irreversible inhibition. Which of the following inhibition is called mechanism based enzyme inhibition? All of you, please try to answer the question, since the no number will be given for answering in the class. So, if your answer is wrong, it does not matter, but please try to answer the question. Okay. Which of the following inhibition is called mechanism based enzyme inhibition? Competitive inhibition, non-competitive inhibition, suicide inhibition, or active site directed irreversible inhibition? I will give you some time, think, and then let me know in the chat box. I will wait for 2 minutes and then I will check the chat box. Let me check the chat box now. Okay, I have few answers. Number C. So I have two number C's for this question number 4. Let me check if number C is the correct answer or not, and let us discuss it. So the enzyme inhibition, that is called the mechanism based enzyme inhibition, is very good. It is number C, suicide inhibition. So what is a suicide inhibition? Inhibitor binds as a substrate, okay, and is initially processed by a normal catalytic mechanism. It then generates a chemically reactive intermediate that inactivates the enzyme through covalent modification. It is called suicide inhibition because the enzyme itself participates in its own irreversible inhibition. So now what does a irreversible inhibitor do? It will bind as a normal substrate to the active site, then it will form something, a particular chemical entity which is very reactive, and it will form a covalent bond with the particular enzymes, then changing its active site, and it is a kind of irreversible mechanism. So it is a special type of irreversible inhibition of the enzyme activity. It is also known as mechanism based inactivation because a particular mechanism is followed in this particular okay, and the enzyme uses the enzyme's own mechanism to inactivate it. That is why it is called the mechanism based inactivation, because the inhibitor, the suicide inhibitor, is using the enzyme's own reaction mechanism to inactivate it. In suicide inhibition, the structural analog is converted to more effective inhibitor with the help of the enzyme to be inhibited. The substrate like compound initially binds with the enzyme, and then first few steps at the pathway are catalyzed. This new product irreversibly binds to the enzyme and the inhibitor further reactions. Okay. So number C was the correct answer. So now problem number 5, it says what is the purpose of having two aspartate residues in aspartyl protease? Your options are number A, 1 aspartate helps the release of the hydrogen ion from the water, make hydroxide ion, and the other aspartate in turn gives stability to the oxyanion that is being generated. Number B, 2 aspartate residues make the active site more flexible. Number C, none of the option A or B are correct, and number D, both A and B are the correct one. What is the purpose of having two aspartate residues in the aspartyl protease? Number A, 1 aspartate helps the release of the hydrogen ion from the water, make hydroxide ion, and other aspartate in turn gives stability to the oxyanion that is being generated. Number B, 2 aspartate residues make the active site more flexible. Number C, none of the options A and B are correct, and number D, both A and B are the correct. What will be the correct answer for problem number 5? Let me know in the chat box. Others also please participate. Only 2 or 1 to 2 people are answering, but I want all of you to answer the questions. Let me check the chat box. Okay, I have two answers again, number A and number D. So, I have two different answer right now. Let me check which of the following is the correct answer. So, the correct answer is number A. 1 aspartate helps the release of hydrogen ion from the water, make hydroxide ion, and other aspartate in turn gives stability to the oxyanion that is being generated. So, in the members of the same family is a evolutionary related is a proteases. So, there are different proteases like serine proteases, like trips. The example of serine proteases are the trypsin, chymotrypsin, elastase, thrombin, staphylococcus, factor D, factor B, etc. There is aspartate protease like gastrexin, cathepsin D, renin, pepsin, chymosin, HIV protease. We have metalloproteases where the metals are present in the active site like thermolysin, carboxypeptides, matrix metalloprotease, and we have cysteine proteases, we have papain, we have calpain and USP2S. So, these are the example of different proteases. So, what happens, as the name suggested, serine proteases have the serine as the main crucial amino acid in the active site which acts as a nucleophile. In aspartate proteases or aspartyl proteases, we have aspartate residues. In metalloproteases, the metal acts as a catalyst, and then in the cysteine proteases, the cysteine residue, it acts as the main component. Okay. So, what does it happen in the aspartyl protease? So, we have two aspartic residues, one is aspartate and one is aspartic acid. So, what does this aspartate do? It is taking up a proton from the water, making it a hydroxyl ion. So, now this hydroxyl ion is the more potent nucleophile. It will attack the carbonyl carbon of a peptide bond, and a tetrahedral intermediate is forming, and then this particular aspartate again it will take up the proton, okay, and it will give the stability to another this aspartate, it will give stability to the oxyanion that is formed. This oxyanion is getting stabilized with this aspartic acid, and that is how this particular two amino acid residues acts in a aspartyl residues. Okay. So, option A is the correct one. So, now I will go to the problem number 6. It says that inhibition of cytochrome P450 by tianilic acid is an example of dash inhibition. Your options are number A competitive inhibition, number B non-competitive inhibition, number C suicide inhibition, and number D active site directed irreversible inhibition. The inhibition of cytochrome P450 enzyme by tianilic acid is the example of which kind of inhibition? Your options are competitive inhibition, number B non-competitive inhibition, number C suicide inhibition, and number D active site directed irreversible inhibition. Again I am giving you 2 to 3 minutes, try to answer the question. Let me check the chat box. Okay, so I think I have different answers, like it is a question mark. I don't know what is a question mark here. Okay, so number C is the given answer. Okay, let me check if number C is the correct one or not. So, the inhibition of cytochrome P450 by ternalic acid is an example of number C, suicide inhibition. So, suicide substrate are the agents which are converted to a highly reactive species when they undergo an enzyme catalyzed reaction. They form covalent bonds to the enzyme and inhibit it irreversibly. In some cases, it can cause toxicity. For example, diuretic agent ternalic acid had to be withdrawn from the market because it was found to act as a suicide substrate for the cytochrome P450 enzyme involved in the drug metabolism. Unfortunately, the metabolic reaction carried out by this enzyme converted ternalic acid to a thiophene sulfoxide, which proved highly electrophilic. This encouraged a Michael reaction leading to alkylation of thiol group of the enzyme's active site, and loss of water from this thiol sulfoxide restored the thiophene ring and resulted to the formation of a covalent link to the enzyme, thus inhibiting the enzyme irreversibly. So, this is the mechanism. Okay. So, this is the cytochrome P450 which have this thiol group and this thiol group from the enzyme's active site, and this is the structure of the ternalic acid which has a thiophene. This thiol group is encouraging a particular is engaging in a Michael type of reaction, addition reaction with this ternalic acid, and upon the oxidation, okay, and it is subsequently forms a irreversible covalent bond with the cytochrome P450, thus inhibiting the cytochrome P450 irreversibly. And since it is a suicide kind of inhibition, so that is because this ternalic acid is acting as a suicide substrate, okay, and it is using the enzyme's own mechanism to inhibit it. So, that is why it is an example of suicide inhibition. So, now I will move toward the next problem. Problem number 7 says, which of the following is known as antigenic determinant? Your options are number A hapten, number B epitope, number C enzymes, and number D paratope. Which of the following is known as antigenic determinant? Hapten, epitope, enzymes, or paratope? All of you try to answer the questions. Which of the following is known as antigenic determinant? Hapten, epitope, enzymes, paratope? Which of the following is known as antigenic determinant? Hapten, epitope, enzymes, or paratope? Try to answer the questions and let me know in the chat box A, B, C, or D which of the following you think is the correct one. I think all of you know the answer and you can answer it correctly because it is very easy. Okay, let me check the chat box. Okay, number B is the given answer. Let me check if number B is the correct one or not. So, the correct answer will be, yes, it is number B, epitope. So, epitope is known as antigenic determinant. So, let us understand what are all the terms are referring to. So, hapten, a hapten is a molecule that is too small to stimulate the antibody formation by itself. However, when the hapten is combined with a larger carrier molecule, usually a serum protein, the hapten and its carrier together form a conjugate that can stimulate an immune response. So, this is the hapten, which is the very small molecule which do not have any activity of its own, but it can stimulate a larger carrier molecule like a serum protein, and this particular thing can, this conjugate, it can stimulate an immune system. So, what is abzyme? So, definition of an abzyme is that it is a combination of antibody and enzyme, and also it is called known as the catalytic antibody. It is a monoclonal antibody with a catalytic activity. So, this is the structure of a abzyme. We have there here one side it is a tumor cell binding site, one side it is a product activation site, okay, which can be used to find the anti-cancer drug if it is using for cancer. This is the antibody domain and it is the abzyme domain. Then we have a epitope, and epitope, also known as antigenic determinant, is a part of an antigen that is recognized by the immune system, specifically by antibodies, B cells or T cells. Paratope, a paratope is a part of antibody which recognizes the antigen and antigen binding site of the antibody. It is a small region of 15 to 22 amino acid of the antibodies region, and it contains the antibodies heavy and light chains. So, let us see the diagram about the epitope and paratope. So, this is an antibody. In the antibody we have a paratope, the area where the epitopes of the antigen bind. So, this antigen antibody complex, then it will induce an immune response. Okay. So, epitope is so remember it is an antigenic determinant which is a part of an antigen, and it is recognized by the antibodies, and in the antibody there is a paratope which is a 15 to 22 amino acids specific site which will bind with the particular epitope of antigen. Okay. So, the correct answer for antigenic determinant is the epitope, number B. So, now I will go to the next question. So, problem number 8 says a student is conducting an experiment in which he adds an inhibitor to an enzyme catalyzed reaction. When the student first adds the inhibitor, the reaction rate decreases. However, he can return to the reaction rate to normal by adding a large quantity of substrate. What type of inhibitor the student is using? Your options are number A non-competitive inhibitor, number B a competitive inhibitor, number C an uncompetitive inhibitor, number D the student likely made a mistake, the result is unacceptable regardless of any inhibition. So, read the question very carefully. Again I will repeat, it says a student is conducting an experiment in which he adds an inhibitor to an enzyme catalyzed reaction. When the student first adds the inhibitor, the reaction rate decreases. However, he can return the reaction rate to normal by adding large quantity of substrate. What type of inhibitor the student is using? Your options are number A non-competitive inhibitor, number B competitive inhibitor, number C uncompetitive inhibitor, and number D the student likely made a mistake, the result is unacceptable regardless of any inhibition. Read the question carefully, and I think all of you now can answer this question very easily and correctly, because this type of thing I have discussed earlier also. If you read the question very carefully, if you have followed the today's lecture, you can answer this correctly. Read and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 8. A student is conducting an experiment in which he adds an inhibitor to an enzyme catalyzed reaction. When the student first adds the inhibitor, the reaction rate decreases. However, he can return the reaction rate to normal by adding a large quantity of substrate. So, what type of inhibitor is he using? Let me check the chat box. I think I have got 3 answers. Number B is said to be true from all the students. Let me check if number B is the correct one. Yes, number B is the correct one, a competitive inhibitor. So, as I have already discussed that a competitive inhibitor enzyme can bind and forming enzyme substrate complex or enzyme inhibitor complex, but not both, means it cannot form a enzyme substrate inhibitor complex. Competitive inhibitor resembles the substrate and it can be relieved by increasing the substrate concentration. So, as you can see, this is the graph, as you can see. Okay. So, in case of the particular, this is the red one is the inhibitor and the blue one is the normal enzyme catalyzed reaction. So, when we increase the substrate concentration, what will happen? The increase in substrate concentration, the Vmax became same in case of the enzyme catalyzed reaction. Okay. And this is the example of competitive inhibitor is the succinate for the succinate dehydrogenase. Okay, the malonate can also act as an inhibitor because it resembles the succinate, which is the normal substrate for the succinate dehydrogenase. When malonate binds, it the reaction does not form, means the succinate does not convert to fumarate, but if you increase the amount of succinate in this particular reaction, what will happen? The inhibition, it will reverse back. Okay. So, the competitive inhibitor was the correct answer. So, now I will go to the next problem. Problem number 9 says which inhibition study deals with the decrease in the both the Vmax and the Km with the increase of the inhibitor concentration. Your options are number A competitive inhibitor, number B non-competitive inhibitor, number C uncompetitive inhibitor and number D suicide inhibitor. Which inhibition study deals with the decrease in the value of both Vmax and Km with increase in the inhibitor concentration? Competitive, non-competitive, uncompetitive or suicide inhibition? Try to answer the question. I will give you some time and then I will show you the correct answer. Which inhibition studies deals with the decrease in both the value of Vmax and Km with the increase of inhibitor concentration? Competitive, non-competitive, uncompetitive or suicide inhibition? Which inhibition study deals with the decrease in the value of both Vmax and Km with the increase of inhibitor concentration? Let me check the chat box first. Okay, I have, I think, 1, 2 answer. Number C is the given answer. Let me check if number C is the correct one or not. So the correct answer will be, very good, number C, the uncompetitive inhibition. So these are the Lineweaver-Burke plots for the inhibition study. These plots are very important for you to remember. Okay, so first one is the competitive inhibition plot. So as you can see, the red one with the inhibitor and the blue one without the inhibitor. So in case of the competitive inhibitor, what happens that as you can see, both the lines are cutting the y axis in the same point. The y axis in the particular Lineweaver-Burke plot is associated with the 1 by V and the x axis with 1 by S, right. So in case what happens, let me use the pen here. So here the intercept, so we know that in the Lineweaver-Burke plot the intercept is 1 by V max and the slope is Km by V max. So in case of competitive inhibition, as you can see, since the intercept is same, as you can see the intercept, it is the same. Okay, so with the inhibition what will happen, 1 by V max is the same. With inhibitor or without inhibitor means V max is remaining unaffected, but as you can see the slope changes, right. In case of the inhibitor, the slope is much higher, it is the much steeper, since the slope means Km by V max. So the slope is the Km by V max. Okay, so if the V max is unchanged and the slope is increasing, means the Km is increasing. So if the Km is increased and V max is unaffected. What happens in case of the uncompetitive inhibition, as you can see, the slopes are same. The slopes are same, but the intercepts are now changed. So now intercept of the particular with no inhibitor is the smaller one, the intercept is the much higher in case of with inhibitor. So if 1 by V max is the intercept means 1 by V max, right. So if the 1 by V max with inhibitor, it is with inhibitor, I will say, since with inhibitor 1 by V max is greater than 1 by V max without inhibitor. So what does it means? It means that the V max is getting reduced, okay, and since the slope is getting increased, so if the V max and the slope is the same, means the Km will also reduce. In case of the particular non-competitive inhibitor, what will happen? The Km will remain unaffected here. As you can see, if you extrapolate this line, it will cut the negative x axis over the same point, means 1 by minus 1 by Km value is the same, means the Km will remain unaffected. But since the particular our intercept is decreased, or sorry, the intercept is increased with inhibitor. So what will it means? It means that the V max is reduced. So these three things are very important. These three graphs are very important. You have to remember the graphs, and if you can remember the graph, you can easily answer these type of questions. Okay. So option number C was the correct answer. So now I will go to the next problem. So the problem number 10, it says that which of the following statement is false regarding ASDII. ASDII means active site directed enzyme inhibitor mechanism, enzyme inhibitor. Okay. So this is the options A is the TPCK is an inhibitor of chymotrypsin. Number B, TLCK is an inhibitor of trypsin. Number C, the inhibitors do not have any directional property. And number D, the reaction functionality of the inhibitor is responsible for this kind of mechanism. Which of the following statements is false regarding the ASDII? Again I will repeat the options. Number A, it says TPCK is an inhibitor of chymotrypsin. Number B, TLCK is an inhibitor of trypsin. Number C, the inhibitors do not have any directional property. And number D, the reactive functionality of inhibitor is responsible for this kind of mechanism. I am giving you some time, think and then answer. which of the following statements is false regarding ASDII? Let me check the chat box. Okay, I have multiple answers, I think. Okay, I have only one answer. It is saying number A and then number C. Okay, A or C, both can be potential answers. Let me check. So, the correct answer will be number C. The inhibitors do not have any directional property. This statement is false regarding ASDII. So, active site mapping via irreversible inhibitors, okay, or it is also known as active site directed irreversible inhibitors. So, this is an example, okay, the TPCK is an example, this kind of a particular example for this ASDII for chymotrypsin. So, this is the natural substrate for a chymotrypsin, right. So, this is a chymotrypsin. Here we have a histidine 57 which is important, and this is the tosyl L phenylalanine chloromethyl ketone, which is known as TPCK. So, this is a ASDII kind of inhibitor for the chymotrypsin, okay. This is also known as TPCK. So, what does this do? It will it has a side chain which is reactive, and this reactive side chain binds with the histidine 57 is subunit of this chymotrypsin, and inhibits this particular enzyme irreversibly, okay. And since it is a particular active site directed mechanism, so that is why it has a directional property. So, the statement number C is false, okay. So, TPCK is an inhibitor of chymotrypsin, TLCK is another example which is an inhibitor of trypsin, and the reaction functionality of the inhibitor is responsible for this particular kind of mechanism. So, remember that TPCK is an active site directed irreversible inhibitor of this particular chymotrypsin enzyme, okay, and this is the structures. If you can remember the structure, it is also very good, but if you do not remember it, it is fine, just remember the name of the particular inhibitor, okay. So, now I will go to the next problem. So, it is a particular mathematical question. I will give you more time for these questions. The turnover number of chymotrypsin is 100 per second, and for DNA polymerase it is 15 per second. The velocities of the reaction catalyzed by both enzyme at saturating substrate level could be made equal if dash times for more DNA polymerase than chymotrypsin were used. Fill in the blanks. The options are number A 1500, number B 0.115, number C 67, and number D 6.7. The turnover number for chymotrypsin is 100 per second, and for DNA polymerase it is 15 per second. The velocities of the reaction catalyzed by both enzymes at saturating substrate levels could be made equal if dash times more DNA polymerase than chymotrypsin were used. Fill in the blanks. You have to fill in the blanks. This is a mathematical question, you have to solve it. I will give you some time to think and to try if you can solve this kind of questions or not. turnover number, what is a turnover number? I have discussed in the my previous lecture. Today I will also give you an overview about the turnover number through this question. Let me check the chat box if there is any answer. Okay, I have few answers like number D and number B. Okay, let me check if the correct answer is number B or D, or it is something else, and let us discuss it. So, the correct answer will be number D, 6.7. Very good. Now, let me show you how to solve this kind of question. So, basically turnover number and maximum velocity. So, turnover number is the unit is per second or second inverse is defined as the number of the product molecule formed by each enzyme active site per second. So, think the kcat as a frequency of catalysis and number of catalytic cycle per second. The value of kcat is the measure of catalytic efficiency. The fastest the enzymes have the highest kcat value. Only kcat is the true constant. Its value depends on the enzyme's catalytic power. And note that the Vm cannot be a true constant. The value of Vm depends on the enzyme concentration. So, now let me show you how to solve it. So, this is the Michaelis-Menten equation. Okay, at the high substrate concentration, where the particular substrate concentration is much greater than Km, the equation looks like this. Okay, the rate equal to K2 E0 equal to Vmax. That is the rate attains the maximum value and the reaction is 0 order with respect to the substrate concentration. This K2 is called the turnover number. It represents the number of molecule is converted to the product per unit per time of the molecule. Now, let us assume that Kc is the Xc is the strength of in molarity of chymotrypsin enzyme, and Xd is the that of the DNA polymerase. Okay. So, now if we can use this equation, so we can say that Vmax of chymotrypsin equal to 100 into Xc, 100. Why? Because it is the turnover number, the K2. If you use this equation, K2 is the turnover number, we are just replacing the values. Okay. So, Vmax of chymotrypsin will be K2, the 100 is the particular turnover number into Xc, which is the strength in molarity, and Vmax of DNA polymerase is 15, the 15 is the turnover number for DNA polymerase into Xd, that is the strength of the DNA polymerase, right. If the velocities of the reaction both has to be same at the saturated substrate level, so what will happen? The Vmax of chymotrypsin will be equal to Vmax of DNA polymerase. If you equal these two side, what will happen now? If you can make the, and if you can change the side, you can see that the DNA polymerase concentration divided by chymotrypsin concentration will be 100 by 15 means 6.7. That is, the concentration of DNA polymerase has to be 6.7 times more than that of chymotrypsin if the both velocities of the reaction has to be same at the saturated substrate levels. Okay. So, that is how you have to solve this kind of problem. I hope it is clear. When I will share this presentation, please go through this kind of all the mathematical questions, okay, and try to solve the questions. I think it will be okay if you if when I share you the PPT, you practice this by yourself also. So, number D was the correct answer. So, now I will move toward the next problem. Problem number 12, it is again a mathematical question. Consider the following plot of the velocity versus substrate concentration of the enzyme catalyzed reaction obeying Michaelis-Menten kinetics with an initial enzyme concentration of 2 into 10 to the power minus 5 micromolar. The turnover number for the reaction is 8000. Number A, number B 8 lakhs, number C 40,000 and number D 4 lakh. This is the graph which you have to use to solve this question. Okay. In the y axis we have constant the Vmax or V with the unit of micromole per minute, and in the x axis we have the substrate concentration in molar. So, you have to calculate, now you have to calculate the turnover number. The initial enzyme concentration is given as 2 into 10 to the power minus 5 micromolar. I will give you some time if you have use a pen and paper, and you can use the graph to solve this question. Consider the following plot of velocity and substrate concentration graph. Initial concentration is given, you have to calculate turnover number. You can use the previous equation which I have shown you to calculate this particular answer. So, what is the turnover number for the reaction? You have to identify. Let me check the chat box. Let me check the chat box if there are any answers. Okay, I think I have few answers. Number A is the only answer I can see here. Okay, let me check if number A is the correct one or not. So, the correct answer will be number D. It is 4 lakh minute inverse. So, now let me show you how to do it. So, as I have shown you the equation earlier, this is the Michaelis-Menten equation. The V0 equal to K2 S0 E0 divided by S0 plus Km. At high substrate concentration, what happens? We can neglect the Km. Okay, at the high substrate concentration we can neglect the Km. So, the equation look like this: Vmax equal to K2 E0. Here rate attains a maximum value and reaction is 0 order. Here K2 is called the turnover number, which represent the number of molecule converted to product in unit time by one molecule of enzyme. So, according to the question, now E0 is given 2 into 10 to the power minus 5, okay, and the Vmax is 8 micromolar per minute. As you can see here, if you just join these lines, this is the Vmax, this is the V, and if you just extrapolate and it joined on the y axis, we will get the value 8. So, Vmax is 8 micromole per minute, the unit is given here. So, now the K2 will be what? The K2 will be Vmax by E0 means 8 micromolar divided by 2 into 10 to the power minus 5 micromolar minute inverse, which gives us the particular result. Okay. So, now I think it is clear. If you have any doubt, you can let me know. Is it clear or not? Let me know if there is any doubt. Anyway, when this type of mathematical questions, sometimes it is difficult to understand it during the ongoing class, but if you check the PPT when I will upload it, I think you will understand the questions. Okay. If not, you can just mail me, I will repeat the question in the next class also. Okay. So, now I will move toward the next problem. So, the next problem is, it is again a mathematical question. Consider the following plot of 1 by V0 versus 1 by S plot of Lineweaver-Burke plot. The result are obtained from the graph is the intercept value is 0.4 micromolar minute inverse and the slope is 7 into 10 to the power 5 minute. What is the Km value of the reaction? I have already discussed the Lineweaver-Burke plot, what is the slope and what are the slope and what is the intercept, and I have discussed it earlier when I have shown you the graph. So, now just you have to put the values and you can calculate the Km. It is easy, I think it should not take much time, but I will give you some time. So, try to answer the question. Options are number A 2.8 into 10 to the power 5 micromolar, number B 0.057 into 10 to the power 5 molar, number C 17.5 into 10 to the power 5 micromolar, and number D 0.11 micromolar. The Lineweaver-Burke plot, intercept is given and slope is given. So, now you have to identify what is the Km value. So, whenever you are studying about the enzyme kinetics, make sure that you remember all the graphs and all the values of the slopes and intercept, because many questions can be given from this particular graphs. If the slope and intercepts are known and the values are given, you can calculate any value for the particular plot. Let me check the chat box now if there are any answers. I think no. Okay, fine, I will show you how to solve it. So, the correct answer will be number C, 17.5 into 10 to the power 5 micromolar. Let me show you. So, this is the Lineweaver-Burke plot. This is also known as a double reciprocal plot. So, if the question is given like calculate this particular values from a double reciprocal plot, you have to understand that it is a Lineweaver-Burke plot. How this particular plot is coming, I have discussed in the previous class slide. So, you can go through the week 2 presentation and then you will find it. So, this is the Lineweaver-Burke plot. Okay, we in the y axis we have 1 by v, on the x axis we have 1 by s, and this is the straight line. We have the slope is km by v max and the intercept is the 1 by v max. So, as the question is given that the Lineweaver-Burke plot, this is the equation 1 by v0 equal to 1 by v max plus km by v max into 1 by s. Here if we plot 1 by v0 versus 1 by s, we will get the following information. The intercept is 1 by v max and the slope is km by v max. Now, as the equation is given, 1 by v max is 0.4 micromolar per minute, okay, means v max is 2.5 micromole per minute. So, and the slope, it is your km by v max, the slope. So, the km is the slope into v max, okay, and the slope is given as 7 into 10 to the power 5 minute, and the v max is coming as a 2.5 micromolar per minute, right. So, our answer km is 17.5 into 10 to the power 5 micromolar. So, see, this is very easy. If only you have to put the values and you will get an answer. Anyway, you do not have to worry, because I will share the PPTs, and I am very sure that if you go through the slides and if you practice it once, you will easily, you will be easily question this kind of problems. So, now problem number 14, it says that consider the plot of the concentration of substrate by V0 versus concentration of substrate plot of the Hanoi Wolf. So, this is another kind of enzyme catalyzed reaction plot like Lineweaver-Burke plot. It is known as the Hannes Wolf plot. The results, so this is the y axis, we have substrate concentration by V, and in the x axis we have substrate concentration. So, the results from the graph is the intercept value is 0.5 and the slope is 0.0001 millimole inverse. What is the km of the reaction? Everything is given in this particular plot. The intercept is km by v max and the slope is 1 by v max. You have to calculate what is the km. Your options are number A 5000 millimolar, number B 500 millimolar, number C 2500 millimolar, and number D 250 millimolar. Consider the plot and answer the question. Try to solve this question. Everything is given right now. You just have to calculate it. Let me check the chat box. Okay, I have one answer which says it is number D. Let me check if number D is the correct answer or not. So, the correct answer will be It is number A. Okay. Now let me show you how to do it. So as you can see in the Hannes-Wolff plot, the equation is this: substrate concentration by V0 equal to substrate concentration by Vmax plus Km by Vmax. Now if we plot a substrate concentration by the concentration of V0, okay, versus or sorry, it is not concentration, it is the V0 versus substrate concentration, we will get the following information, that the intercept will be Km by Vmax and the slope is 1 by Vmax. Now from the equation it is said that Km by Vmax, the intercept value is 0.5, right, and 1, so 1 by Vmax is the slope value, is the 0.0001 millimole inverse. So now Vmax will be 10,000 millimole if we just make it reciprocal, and Km will be here 0.5 into Vmax. So if Vmax is 10,000 millimolar, so Km will be 0.5 into 10,000 is equal to 5,000 millimolar. So that is how this type of question you have to answer, you have to solve. So now I will go to the next problem. So problem number 15 says, which of the following statement is incorrect? Your options are: number A, production of monoclonal antibody is expensive; number B, polyclonal antibodies are produced by the same clone of plasma B cells; number C, production of polyclonal antibody does not require hybridoma cell lines; and number D, monoclonal antibodies are used as therapeutic drugs. Which of the following statement is correct? Number A, production of monoclonal antibody is expensive; number B, polyclonal antibodies are produced by same clone of plasma B cells; number C, production of polyclonal antibodies does not require hybridoma cell lines; and number D, monoclonal antibodies are used as therapeutic drugs. Which of the following statement is correct? Production of monoclonal antibody is expensive. Number B, polyclonal antibodies are produced by same clone of plasma B cells. Number C, production of polyclonal antibodies does not require hybridoma cell lines. And number D, monoclonal antibodies are used as therapeutic agents. Let me check the chat box. Okay, I have an answer. It is saying it's number C. Let me check if number C is the correct one or not. So the correct answer will be number B. Polyclonal antibody are produced by same clone of plasma B cells. This statement is incorrect. So what is a monoclonal antibody? So these are antibodies that are identical because they were produced by one type of immune cell or B cell, all clones of a same parent cell. Polyclonal antibody represent antibodies from multiple clones of B lymphocytes and therefore bind to a number of different epitopes. For example, for immunoglobulin. So these are the differences between the monoclonal and polyclonal antibodies. Monoclonal antibodies refer to a homogeneous population of antibodies that are produced by a single clone of plasma B cells. Polyclonal antibodies refer to a mixture of immunoglobulin molecules that are secreted against a particular antigen. Monoclonal antibodies are produced by the same clone of plasma B cells. Polyclonal antibodies are produced from different clones of plasma B cells. That is why number B statement is the incorrect one. So production of monoclonal antibody requires hybridoma cell lines. Production of polyclonal antibody does not require hybridoma cell line. A homogeneous antibody population is the monoclonal ones, but the polyclonal ones are heterogeneous antibody population. In the monoclonal antibodies interact with a particular epitope of antigen, but the polyclonal antibodies interact with the different epitopes of the same antigen. The production is expensive in case of monoclonal antibody, the production is not expensive for the polyclonal antibody. The skills are required to handle the technology for monoclonal antibody production, fewer skills are required for the production of polyclonal antibody. The monoclonal antibody production takes some time, but the polyclonal it takes less time. This possesses less cross reactivity, the polyclonal one possesses comparatively high cross reactivity. The monoclonal antibody used as therapeutic drugs, polyclonal antibodies are general research application. Advantages of monoclonal antibody include immortal supply, high specificity and high reproducibility. The advantages include high affinity, tolerance of minor changes and more robust detection in case of polyclonal antibody. Okay. So, these are the differences between monoclonal antibody and polyclonal antibody. So, now I will go to the next problem. Problem number 16 says, what is the use of hybridoma technology? Your options are: number A, separation and isolation of monoclonal antibodies from polyclonal set; number B, separation and isolation of lymphocytes from immunoglobulins; number C, separation and isolation of haptens from epitopes; and number D, separation and isolation of septum for enzymes. What is the use of hybridoma technology? Number A, separation and isolation of monoclonal antibody from polyclonal set. Number B, separation and isolation of lymphocytes from immunoglobulins. Number C, separation and isolation of haptens from epitopes, and number D, separation and isolation of haptens from enzymes. Let me check the chat box now. So, the answers for problem number 16. Okay, I have one answer which says it is number A. Let me check if number A is the correct one. So, the use of hybridoma technology is very good. Number A, separation and isolation of monoclonal antibody from a polyclonal set. So, hybridoma technology is a method for producing large number of identical antibodies called monoclonal antibodies. It was discovered by Kohler and Milstein in 1975. They were awarded Nobel Prize in Physiology and Medicine in 1975. The hybrid cells are produced by fusing B lymphocyte and myeloma cells or tumor cells. The B lymphocytes have ability to produce large number of antibodies and tumor cells have the indefinite growth. That is why two cells are used to production of the hybrid cells. So, monoclonal antibody, as I have already discussed, that it react with a specific epitope on a given antigen, but polyclonal antibody react with various epitopes on a given antigen. and this is the hybridoma technology in short. So, we have the B cells which are isolated, and then we have the myeloma cells, then the fusion of B lymphocyte and myeloma cells, the culture in HAT media, HAT media, and the test each wells for specific antibodies and screening of the hybridoma cells. It gives as the expansion of positive clones which and the propagation in vitro and in vivo gives us the monoclonal antibody. So, this is a basic chart for this thing. Okay. You just have to remember what is the hybridoma technology, what is the hybridoma cells, and what is the application. So, now I will move toward the next problem. So, problem number 17, again it is a mathematical question. So, consider the following values of Michaelis-Menten enzyme kinetics and calculate the dissociation constant for an enzyme substrate complex and catalytic efficiency for the enzyme. The K1 is 4 into 10 to the power 8 molar per minute inverse. K minus 1 is 1 into 10 to the power 3 minute inverse, K2 is 6 into 10 to the power 3 minute inverse. You have to use this 3 constant value to calculate the dissociation constant for enzyme substrate complex and catalytic efficiency of the enzyme. Your options are number A 0.5 micromolar and 8.57 10 to the power 8 molar minute inverse, number B 2.5 micromolar into 8.57 into 10 to the power 8 molar minute inverse, number C 2.5 micromolar and 0.857 into 10 to the power 8 molar minute inverse, and number D 5 micromolar into 10 to the power into 0.857 10 to the power 8 molar minute inverse. So, these are from this set you have to identify the correct set for the dissociation constant of enzyme substrate complex and catalytic efficiency. So, I know you know what is the K, K minus 1 and K2. I hope that you know all these things. If you follow the Michaelis-Menten kinetics, you will know the enzyme and substrate. First it forms the enzyme substrate complex and then it dissociates into the enzyme and the product. So, the enzyme, the first step is the reversible one, the forward constant is K1, and for the reversible the backward is the K minus 1, and for the second step the constant is K2. I will just draw it here, so that you can understand. So, we have a enzyme, right, plus we have substrate. So, first step is the reversible step. Here it will form what? It will form a enzyme substrate complex like this, and then this step is forward direction only. So, it will have a enzyme, we will have a enzyme. Okay, this is all the considers as the concentrations, and plus we have product. So, for this first step, the step 1, the forward rate constant is small k1 and the backward rate constant is k minus 1, and for the second step the forward rate constant is the only rate constant which is the k2. These 3 values are given. You have to identify the dissociation constant for this enzyme substrate constant complex and the catalytic efficiency of the enzyme. I will give you some time, try to answer this question. Let us do this mathematical equation. Let me check the chat box. Hello. Hello. If my voice is audible. Hello. Am I audible? The mic is not working. Let me know if I am audible. Any problem with the audio? Okay. Okay. Let me show this problem, how to solve it. Okay. So the correct answer will be number C. 2.5 micromolar is the answer for the enzyme substrate complex dissociation constant, and the catalytic efficiency is 0.857 into 10 to the power 8 molar minute inverse. So basically the dissociation constant for enzyme substrate complex is given by K minus 1 divided by K1. So it will be so the K minus 1 is 1.1 into 10 to the power 3 minute inverse divided by 4 into 10 to the power 8 minute inverse molar inverse minute inverse. So the answer is 0.25 into 10 to the power minus 5 molar, which is 2.5 micromolar, and the catalytic efficiency is given by K2 by Km. Now Km is given by K minus 1 plus K2 divided by K1. If we do all the calculation, you will find the catalytic efficiency is K2 by Km will be 0.857 into 10 to the power 8 molar minute inverse. Okay. Okay, it's not audible. Hello, is it audible now? Let me know if you can hear my voice now. Okay, I will repeat the particular question. Okay, the mic was not working for some time. So as you can see, the dissociation constant for enzyme substrate complex is given by K minus 1 divided by K1. Okay, so now as all the values are given, as you can see, K minus 1 means 1 into 10 to the power 3 minute inverse. So it will be 1 into 10 to the power 3 minute inverse divided by K1, which is 4 into 10 to the power 8 minute molar inverse minute inverse. If you solve the question, you will find, if you calculate, you will find that the answer is 2.5 micromolar. Similarly, the catalytic efficiency is given by K2 by Km. The Km is K minus 1 plus K2 by K1. Okay, and if you calculate, the Km will come as 7 into 10 to the power minus 5 molar. Hence, the catalytic efficiency, which is K2 by Km, it will be 6 into 10 to the power 3 minute inverse divided by 7 into 10 to the power minus 5 molar, which is 0.857 into 10 to the power 8 molar minute inverse. So you can hear me, right? So if you have any problem for listening to the voice, let me know. I will move toward the next problem. So the problem number 18 says, which inhibition has the apparent Km value as Km of the normal enzyme catalyzed reaction? The options are number A competitive, number B non-competitive, number C uncompetitive, and number D mixed. So which inhibition has the apparent Km value same as the Km of the normal enzyme catalyzed reaction? As we know that in the normal enzyme catalyst reaction, there is a Km, which is a Michaelis-Menten constant. But when there is an inhibition, the equation changes and all the values changes for Km, for Vmax, and all the values are known as apparent Km and apparent Vmax. Okay, so here you have to identify in which enzyme, which inhibition the apparent Km value is same as the Km of the normal enzyme catalyzed reaction where there is no inhibition. Means with inhibition and without inhibition, in both cases the Km value is the same. Am I audible? I hope I am audible, right? Okay, I also have one answer, so seems like I'm audible. Okay, I will give you some more time. I will give you one more minute, 1 to 2 more minutes to identify the correct answer. So in which inhibition apparent Km value is same as the Km of normal enzyme catalyzed reaction? So I will show you the answer. Already in the chat box, I think I have one answer which says it is the number A. Okay, let me check if number A is the correct one or not. Okay. So the correct answer is number B, non-competitive. So this is a chart which shows you the different values which are associated with a different kind of inhibitions. Like for competitive, the apparent Vmax is the normal Vmax. This is the value of apparent Vmax by Km, and this is the value of the apparent Km. In the mixed inhibition, these are the values. In non-competitive one, these are the values. So as you can notice, in non-competitive inhibition, the apparent Km is the same as the normal Km, and this is the values for the uncompetitive reactions. This chart, it is better if you can remember this chart, because the questions can come from related to this different type of values of different inhibition kinetics also. Not only you have to remember the normal enzyme catalyzed reactions, but for the inhibition also it is better if you can remember this chart. Okay, so now problem number 19 says, which of the following agent does not have any effect of its own? Your options are number A agonist, number B antagonist, number C partial agonist, and number D inverse agonist. So which of the following agent does not have any effect of its own? Agonist, antagonist, partial agonist, inverse agonist. I will be a little bit faster because we are always almost at the end of our session. So which of the following agent does not have any effect of its own? Agonist, antagonist, partial agonist, inverse agonist. Let me check the chat box. Okay, I don't think. Okay, let me show you the answer and let us discuss a little bit. So agent that does not have any effect of its own is the number B, antagonist. So it is defined. So receptors are the macromolecules. So there are different terms which are we can use as the effector molecules, which can use are used in drug receptor interaction. So agonist is an agent which activates the receptor to produce an effect similar to that of the physiological signal molecule. Inverse agonist is an agent which activates the receptor to produce an effect in the opposite direction to that of the agonist. Antagonist is an agent which prevents the action of an agonist on a receptor or the subsequent response, but does not have any effect of its own. And partial agonist is an agent which activates the receptor to produce submaximal effect but antagonizes the action of the full agonist. Ligand. A ligand is any molecule which attaches selectively to particular receptors or sites. The term only indicates affinity or ability to bind without regard of the functional change. Agonist and competitive antagonist are both ligands of the same receptor. Okay. So now I will show you the last question of today's session. It says, which of the following statements is false regarding antibodies? Number A, B cells are associated with the humoral immunity. Number B, T cells are associated with the cellular immunity. Number C, T lymphocyte differentiate into plasma cell. And number D, B lymphocytes get maturation in thymus. Which of the following statements is false regarding antibodies? Number A, B cells are associated with humoral immunity. Number B, T cells are associated with the cellular immunity. Number C, T lymphocytes differentiate into plasma cell. And number D, B lymphocytes get maturation in thymus. Let me check the chat box if there are any answers. Okay, number D is the given answer. Let me check if it is the correct or not. So, the correct answer will be number D. Very good. B lymphocytes get maturation in thymus. This statement is false regarding antibodies. So what is an antibody? A blood protein produced in response to and counteracting a specific antigen. Antibodies combine chemically with substance which our body recognizes as aliens, such as bacteria, virus, foreign substance in the blood. Antibodies, also known as immunoglobulins, are Y-shaped proteins that produced by immune system to help stop intruders from harming the body. When an intruder enters the body, the immune system springs into the action. These invaders, which are called antigens, can be viruses, bacteria, other chemicals. When an antigen is found in the body, the immune system will create antibodies to mark the antigen to the antigen for the body to destroy. So B cells, they attack the inhibitors from outside the cells, and the T cells, they attack the infected cells itself. So what are the differences in B lymphocytes and T lymphocytes? B lymphocytes origination and maturation takes place in bone marrow. For T lymphocyte, origination in bone marrow but maturation takes place in thymus. B lymphocytes, they differentiate into plasma cell. T lymphocyte differentiate into plasma cell or memory cell or cytotoxic T cells. Now, B lymphocyte involved in humoral immunity. T lymphocytes are involved in cellular immunity. And the B lymphocytes reproduce antibodies, and T lymphocyte, they help B lymphocytes to produce antibodies. Okay. So answer number D was the incorrect, which says that B lymphocyte get maturation in thymus, but B lymphocytes, the origination maturation takes place in bone marrow, whereas for T lymphocyte, origination takes place in bone marrow, but the maturation takes place in the thymus. Okay. So this was the last question of today's session. So now I will quickly share my email ID with you here in the chat box. Okay. So today what we discussed about many things about the enzyme inhibition. Okay, let me first write my email ID. So this is my email ID. If you have any doubt, any query regarding today's session, make sure that you drop an email to my email ID here, or you can also write it up in the discussion forum of the SWAM portal for the NPTEL course of Organic Chemistry and Biology and Drug Development. Today it was our week 3 live session. Here we discussed about the enzyme catalyzed reactions. We did a lot of mathematical questions, and we also discussed about different inhibitions and something about the synthetic biology, antigen, antibodies, and so on. So this lecture PPT and also the recording of today's class I will upload, and you can find it on the SWAYAM portal, as I have already shown you. Now where you have to find it up, you can go to the sports outline where there is a week wise upload. Here you have to click on the problem solving recording, and you have to click here, and then you will find out the lecture PPT of today's class, and also the lecture video, the Google Meet what we had done today. So now I will close the session for today. If you have any doubt, any query, you can ask me now, or you can drop an email to me. I will happy to answer your queries. If not, I can close the session for today. So thank you all for joining the session. We will meet on the next Monday from 6 p.m. to 8 p.m. for the next class. And if you have any doubt, any query, please note the email ID and you can drop an email to me. Okay. Thank you all. I will close the session for now. Let's meet on the next Monday.